Okay, so my name is Alexis Queen, and I did option B for this test video. I wanted to share some background. Marshall Nuremberg wanted to solve how the genetic code was translated. And at this time, people knew that mRNA could be mixed with ribosomes and amino acids to create proteins, but they didn't know which specific nucleotide on mRNA were used to create specific amino acids. So on the top, I drew out the experiments, but in 1961, Nuremberg and Mattei wanted to decipher the genetic code using in vitro translation system. So they had poly-U homopolymer and mixed it with each amino acid individually. They had 20 tubes, 19 not radioactive and 1 radioactive. The radioactivity amino acids actually help because they can track which amino acids are being coded for the strand of mRNA they were putting in. They then incubated the tubes, and then translation occurred. The protein was then filtered and radioactivity was checked. The tube that was radioactive was unlike the rest because a homopolymer coded for the radioactive amino acid. In this case, it was UUU, which coded for phenylalanine. They then repeated this experiment with different homopolymers, and they got that AAA coded for lysine, CCC coded for proline, and GGG, they never actually got to work. So then new technology came around, and Nuremberg and Leader in 1964 wanted to see if they can code for all codons, not just the homopolymers. So they took synthetic mRNA with one codon and tRNAs with amino acids and ribosomes, and they mixed it all together. They then found that the ribosome stuck to the mRNA and the tRNA specified by the mRNA. They then filtered the solution, and the tRNAs paired with ribosomes were bound. mRNA stuck to the filter, and the unbound passed through. The filter was assayed, to which the scientists could then determine which amino acids were bound. They then were able to repeat this and identify 61 of 64 codons, and the other 3 were stop codons. In conclusion, this gave us our genetic coding dictionary, which is mostly universal. I find that this experiments were the basis of genetics.